Thank you for joining us, everybody. This is the May 2024 Seminar on Physical Genomics. My name is Benjamin Keene, here at CPGE at Northwestern. Just a quick word for those of you who are new to this seminar. Center for Physical Genomics is a transdisciplinary research center at Northwestern that studies the structure and function of the genome through the development of new super resolution imaging and nanosensing technologies, modeling techniques, computational genomics, artificial intelligence, and other methods. We've got a mission to develop new strategies for the treatment of disease and the reversible engineering of living systems, and to train the next generation of multidisciplinary scientists and bioengineers. You can learn more about our research and training programs at physicalgenomics.northwestern.edu, or you can follow us on YouTube, Twitter X, or Instagram. We do have an archive of all of our seminars and public lectures on YouTube, so do check that out. We're really happy to welcome our invited speaker today, Professor Steve Jacobsen of UCLA. Dr. Jacobsen is an investigator of the Howard Hughes Medical Institute, professor of molecular and cell developmental biology. His research interests center on mechanisms of epigenetic inheritance in plants, including the genetics and genomics of DNA methylation, histone methylation, and small RNA-driven silencing pathways. This lab also develops tools for gene editing in plants, as well as tools for targeted epigenetics using CRISPR systems. He's published over 220 research articles and reviews. He's received the Beckman and Searle Investigator Awards, and he was elected to the National Academy of Sciences in 2011 and the American Academy of Sciences in 2016. Dr. Jacobsen, it's an honor to have you share your work with us today. Thank you so much for joining us. For our audience, there will be time for questions at the end of the talk, or you can just type them into the Zoom chat when you think of them, and we'll address them at the end. So at this point, Dr. Jacobsen, if you want to get started, I will turn the floor over to you. Thank you so much.
All right. Well, yeah, thank you so much for the kind introduction, and it's a pleasure to speak in this series. You guys, I've known Vadim for a long time. You guys have a lot of really cool stuff going on there. So I'm going to talk about one of the main projects in the lab, studying the mechanisms of gene silencing by DNA methylation, and really highlight the work of two super talented graduate students, Lucia Ichino, who's now a postdoc at Stanford, and Brandon Boone, who's a current graduate student. So we're going to be talking about DNA methylation, which I think most people know is usually a mark of gene silencing, although, as I'll mention, it can also be involved in gene activation. It's basically conserved throughout eukaryotes. It evolved from bacterial systems initially, and plants have a particularly high level of DNA methylation, and cytosines, which are the methylated base, can be methylated in all sequence contexts. We tend to classify them into three buckets: C's followed by G's, CHGs, and CHHs, where H is just not a G. And that's because after years of studying these pathways, we know that the mechanisms by which these methylations are maintained are different depending on the sequence context. So we've been working on these pathways for many years now. We're virtually done working on them because most of the questions have been really answered. So I'm just kind of giving you the essence of them as background information. CG methylation is the most well conserved. It basically works the same in plants and animals. It's essentially a copying mechanism of DNA methylation at the replication fork, and it works because of the symmetry of CG dinucleotides. CG's the same on both strands, 5' to 3', And so after DNA replication, one of the strands will be methylated. The other one is unmethylated. We call that hemimethylation. And there's a component in the system called VIM in plants and UHRF1 in mammals, which directly recognizes that hemimethylated state and recruits the DNA methyltransferase called MET1 in plants and DNMT1 in animals to fill in methylation on the other strand, thereby replicating the pattern that was in the mother cell. So in this way, CG methylation is almost like a fifth base of the DNA that's just is replicated. Now, the other types of methylation are driven by very different pathways. CHG methylation is put on by the methyltransferase enzyme called CMT3. CMT3, after it deposits methylation, recruits a series of proteins. Kryptonite is the first one that we discovered, that can bind directly to methylation. So they have a known methylation binding domain. And these are enzymes that put on a particular mark on the histone tail, H3K9 dimethylation, which then it turns out CMT3 has two motifs that bind to that mark. So the maintenance of this type of methylation is a simple feed forward loop between these two different kinds of modifications. Then there's a third pathway that works basically the same. The only thing is the enzyme is CMT2, a close cousin of CMT3 that prefers CHH sites. And then finally, this very complicated RNA-directed DNA methylation pathway, which is maintaining other of the CHH sites. This is also the pathway involved in initial establishment of methylation. And in part, it can establish at specific loci because it uses small interfering RNAs, 24 nucleotide siRNAs, which are enough of a unique address to tell the pathway exactly where to go by pairing up to the sequence at its locations. And it does that. So there's a whole machinery that produces the 24 nucleotide siRNAs, and then a whole nother set of machinery that produces non-coding transcripts made by a polymerase called Pol5, which serve as the template upon which these Argonaut siRNAs bind. And wherever those two come together, that's the trigger to bring in the DNA methyltransferase, DRM2, that puts on this type of methylation. So now, what's really surprising is that despite knowing so much detail about how these pathways work and how methylation is established and maintained, we know almost nothing about how does methylation actually silence genes. So we really turned our attention to this question about 6 years ago. And the approach that we took, we figured, well, any pathway which is downstream of methylation, must involve some kind of methyl reader proteins. So we did a fishing experiment by taking nuclear extracts from plants and then finding ones that specifically bound to methylated oligonucleotides. And then we've been studying this big list of proteins ever since. The first set of proteins, the ones in the green, we published now in 2018. These turn out to form a complex. There's a couple of methyl readers and then these DNAJ proteins. And surprisingly, what this complex does is it binds to small regions of DNA methylation upstream of genes. But its function isn't to silence the underlying methylated element. It actually acts to stimulate the transcription of the nearby protein coding gene. Now, we don't know exactly why this pathway evolved, but what we've guessed is that this essentially makes it, allows for transposons to live in the promoters of genes with very few deleterious consequences, because usually having methylation in a promoter is repressive. But with this complex, you sort of normalize that and you bring you bring the level of gene expression of the neighboring gene back up. So it's like the transposon, it's almost as if it's not there anymore. And we imagine that the reason that this kind of pathway evolved is so that you would tolerate and you could even promote transposons being in promoters, because transposons are a major way that promoter regulatory elements are shaped over evolutionary time. And that's true in mammals and in plants. It's just one example. In mammals, something like 80% of OCT4 binding sites are have been brought in by line elements, a type of transposon. Now, we are still working on this pathway because we still do not understand the mechanism by which this pathway is actually upregulating the genes. None of the proteins that came from the study really indicated what that would be. So we're doing an Arabidopsis mutant screen right now to look for new factors that are required for this. And hopefully we'll learn how this pathway works. Now, today I'm really going to talk about a couple of other proteins that we have been studying more recently, called methyl binding domain protein 5 and 6. And these are ones that Lucia Echino started studying. So these methyl binding domain proteins, they're homologous to some mammalian ones. For instance, MECP2 is a well-studied one, the MBD domain. And it's a set of 13 proteins, and we're just starting to dig into the function of all these different ones. We did some homology modeling of the MBD domain itself, together with some of the mammalian ones with Xiaodong Cheng. And we believe that there are 2 conserved arginine residues, which are predicted to be the residues that actually contact the methylated DNA, and these should be required for recognition of methylated DNA. And in line with that, Brandon Boone did fluorescence polarization assays and showed that indeed in vitro MBD6, and not shown here, MBD5, binds very specifically to methylated CG oligonucleotides. But when we engineer in these 2 arginine mutations, that is now completely abolished. So these do seem to be directly binding to methylated DNA. Another assay that we did to confirm this is called the DNA curtains assay. And we did this with Cy Redding's lab at UCSF. And the way this works is you take 50 kb lambda DNA, and they're essentially stretched out by the flow of liquid. And then we compared ones that had been pre-methylated at CG sites versus unmethylated controls. And then we wash in MBD6, and you can really nicely see the MBD6 bind to the curtains. And the density of methyl CGs on these curtains nicely goes with the density of MBD6 binding. Another way that we did this is with a technique called DAP-seq, which is where we took our MBD proteins, made them in vitro, and then we gave them Arabidopsis genomic DNA, and then did a binding experiment and then sequenced to see what are all the DNAs that it likes to bind in natural DNA sequences. And we saw something very interesting, which is that there's a tight relationship between the density of CG methylation on DNAs and binding. And now that's when we use wild type DNA. If we then extract DNA from the MET1 mutant, remember I told you that is the enzyme that puts on CG methylation. So in this mutant, there's no more CG methylation. Now you just get, the curve is just flat because it's basically can't bind to anything. And then the final way that Lucia showed this is with chromatin immunoprecipitation sequencing, or ChIP-seq, where she found that both MBD5 and MBD6 bind to high density sites of CG methylation. And when she engineered in the arginine mutations, now it no longer binds. So I just want to make this point because we're going to get back to it in a bit. It didn't bind all CG sites in the genome. For instance, these more sparsely spaced sites we don't really see binding, only to the dense CG sites. And I'm going to get back to why that may be in a bit. So to start to study the biological function of MBD5 and 6, we made mutants and then did RNA sequencing to look at gene expression effects. And what we found is that in the single mutants, we don't see much, and that's because these are redundant genes. They function redundantly. In the double mutant, however, we see an upregulation of lots of sites, and when you go look, they're all basically DNA methylated genes and transposons. We also wanted to see, do the MBD mutants affect the maintenance of the underlying methylation? And the answer was no. So there's really no difference between wild type and MBD5 and 6. So these seem to be strictly readers of the methylation. They're reading it and causing gene repression, but they're not controlling the maintenance of the methylation. One of the key target genes that we noticed is a gene that I'm going to mention later, and we work on a lot, called FWA. This is a gene that functions in flowering time control. It has dense methylation right in its promoter. We see really nice peaks of MBD5 and MBD6. And in the MBD5/6 mutants, we see an upregulation of FWA. In wild type, it's totally silent. And we see that whether we do poly-A-RNA-seq, total RNA-seq, or GRO-seq, which is basically a nuclear run-on sequencing technique that shows you what's happening at the level of transcription. So we know that we're seeing transcriptional upregulation of the target genes here. If Lucia was able to complement all of these gene expression defects with flag-tagged MBD and MBD5 and MBD6 constructs, but when she engineers in these arginine mutations, they now fail to complement, as you'd expect. Okay, so now we're really interested in what is the mechanism of gene silencing here. And to get started on this, we wanted to see what are the other interacting protein partners. So we did an immunoprecipitation followed by mass spec. And I'm going to talk about, first I'm going to talk about this J domain protein that we found, and then later we'll get back to these small heat shock domain proteins. But the first protein, Lucia spent a lot of time on, it's a J domain protein that she renamed as Silencio, which is Italian for silencing, and she's Italian, so she was having fun with this. So in the Silencio mutant, we essentially see identical phenotypes as MBD5/6. The exact same set of genes and transposons are upregulated. Also, by chip sequencing, Silencio chips to all the identical places as MBD5 and 6. And Lucia also did this chip in mutants. For example, MBD5 and MBD6's pattern does not change at all really in a Silencio mutant, but Silencio binding to the genome is completely abolished in an MBD5/6 mutant. So it's very clear that the MBD5/6 are the readers, they're recruiting Silencio, and then Silencio seems to be involved in the silencing. Lucia also did this, I think this is one of the coolest experiments in the paper, this ectopic silencing assay. So this was focused on this gene FWA that I mentioned to you, which is a natural target of the MBD5/6 and Silencio. So they're naturally causing silencing of FWA. Now, it turns out that there are stable, unmethylated epigenetic alleles of FWA. In plants, methylation changes are heritable for many generations. So we have these epialleles in which the methylation has been lost and it does not come back in subsequent generations. So we can work with these overexpression alleles. Then she reintroduced into that a fusion between Silencio and a zinc finger that we designed to bind to the FWA promoter. to see if we could cause silencing now in the absence of DNA methylation. And indeed we could. We got a partial silencing of FWA in this case. Now it turns out zinc fingers, unlike CRISPR systems, which can be very specific, a lot of zinc fingers are not very specific. And we were able to sort of take advantage of that in this case, because there are like 10,000 off-target sites for which there are pretty good ChIP-seq peaks for the zinc finger. And what we found is when we went and looked, wherever there was a zinc finger peak close to the promoter of a gene, we also saw silencing of that in the Silencio fusion protein. So Silencio seems to be able to silence pretty much any gene that you target it to. So, well, then the question became, what is the mechanism of Silencio-mediated gene silencing? And for this, again, we started with an IP mass spec experiment. And when we pull on Silencio, of course, we get MBD5 and MBD6 as expected. But we also noticed about a 10-fold enrichment for 5 HSP70 proteins. And it turns out we hadn't noticed this initially, but you also see that enrichment when you pull down on MBD5 and MBD6. HSP70s are common contaminants, so we just weren't paying attention to it at first. And what we saw is that when we repeated MBD5 and MBD6 IP mass spec in a Silencio background, now you no longer got the enrichment of the HSP70. So it's the Silencio which is recruiting HSP70. Now it turns out this makes perfect sense with what we know about other J-domain proteins. They are cofactors for HSP70s in protein homeostasis pathways. So they are the component that typically will recognize client proteins, bring them to HSP70s, and then HSP70 uses ATP to help properly fold those proteins. So in part, these proteins are involved in keeping proteins from being denatured and keeping them happy and healthy in the cell. Now it turns out there in many J-domain proteins, there's an HPD tripeptide sequence, which is known to be required for the interaction with HSP70, and Silencio had that. So Lucia made a mutation, H94Q, and she showed that this is essentially a null mutation for silencing. Also, when she repeated the IP mass spec with Silencio H94Q, she no longer enriches for HSP70. Now, importantly, when she redid the ChIP-seq with this mutation, there was really no change in the pattern. So Silencio is still being targeted to all the right places, but it can't interact with HSP70 because of this mutation, and it no longer can cause gene silencing. So what that tells us is that the mechanism of Silencio silencing is going through HSP70. And in this mutant, you can't interact with HSP70 and you can no longer cause gene silencing. So that's what we learned so far. We're going to get back to what, because this is a little bit of a weird mechanism. Why would you recruit these, protein folding chaperone proteins? How does that cause gene silencing? And, when we published this a few years ago, we honestly had no idea, but it was just so novel. We were really motivated to keep working on this. And I'll get back to what we think it's doing in a bit. The key to figuring that out turned out to be studying the other proteins in the complex I mentioned, these small heat shock domain proteins that were also pulled down by MBD5 and 6. So these proteins are called ACD15 and ACD21. And no matter which of these 5 proteins you pull on, you get tons of peptides for all of them. So they are in a stable complex. So what are these ACD proteins? It stands for alpha crystalline domain proteins. Some of the famous ones are like eye alpha crystallins that are involved in keeping your eye from getting cataracts and keeping the fluidity. They're also called small heat shock domain proteins. So when we do chip with ACD15 and 21, again, they chip to all the identical places as MBD5, 6, and Silencio. And the ACD single and double mutants have nearly identical changes in gene expression as the other mutants. So these are required components of the silencing system. Now we did a ton of work to try to figure out what does this complex look like. And I'm not going to take the time to go through all this. It's published in Brandon's paper from last year. But essentially, one of the key experiments is we did IP mass spec of each of these components in mutants of every one of the other components. We also did nuclear localization of each component in mutants of every single one of the other components. And then we used this amazing AlphaFold multimer to make predictions, and the predictions were very strong for each of these interactions that I'm going to mention to you. So the way we think the complex is arranged is MBD5 and 6, remember they're redundant proteins, so it can be either of them. They basically have two domains, the methyl binding domain, which binds methylated DNA, and then this sticky C domain. Please remember this because we're going to be using this domain later. This is the domain that interacts with ACD15. ACD15 then interacts with ACD21, which then interacts with this domain we've called the ZBT domain of Silencio, and then the J domain of Silencio interacts with HSP70. Now, AlphaFold multimer also strongly predicted that there is at least a dimer of dimers, if not multimers of ACDs at the core of this complex, which we hadn't been thinking about. But once we saw this from AlphaFold multimer, it immediately explained why this complex is localized to only to high density CG sites. And the way we would explain this is that since you've got multiple MBD domains per complex, it's only going to, if cooperative binding is needed to adjacent sites, that could really nicely explain why you only see binding to the high density sites and not these more sparsely distributed sites. Now, consistent with that hypothesis and the complex configuration, if we mutate Silencio, so get rid of Silencio, we really don't see much change in the pattern of MBD5 and 6, and that makes sense. There would still be two MBDs, or at least per complex. But if we mutate the ACD proteins, now what we see is a dramatic loss of binding. We still see a little bit of binding to the right spots, but we lose this very nice correlation between methyl CG density and ChIP-seq signal that you see in the wild type. It becomes much flatter. Also, when we mutate this little sticky C domain, we get the same exact effect. I'm not showing the data here, but that makes sense because the sticky C is needed to recruit the ACDs. Okay, now the other approach we took is to localize these components in nuclei. And what we see is that they form these very clear and discrete nuclear bodies when we do confocal live cell imaging. And so these are all fluorescently tagged proteins driven by their natural promoters, and they complement mutant phenotypes, so they're fully functional. And what we see is that they localize to what are called the chromocenters. The chromocenters are dense areas of DNA methylated chromatin. They can be stained with DAPI, which is a DNA stain. And we see that all of the components are localizing there. So here's MBD6 co-localizing with DAPI. Here's ACD15 together with MBD6, 21 with MBD6, and Silencio with MBD6. So, and it makes sense. They're all in the same location, and they're all at these discrete nuclear bodies. And that localization pattern allowed us to do another type of experiment called fluorescence recovery after photobleaching, or FRAP, which allows you to look at sort of the mobility of complexes in a certain area of the nucleus. So the way it works is you use a laser to zap a certain spot, which denatures the GFP So you don't see the spot anymore. And then you look at how fast does that GFP return, which that would have to sort of diffuse in from the surrounding areas in order for GFP to reconstitute a nuclear body. And what we see is that MBD6 RFP recovered to nearly 100% fluorescence with an estimated half-time of about 3.5 seconds, which is as fast as any protein in the literature, I think, for these chromatin proteins. So it's very mobile. But in a Silencio mutant, we saw there was a significant fraction that didn't recover. As an example here, we blasted this spot here in the wild type, and you can see it returns right away. Whereas in Silencio, we blasted this spot and it hardly returns in the time of the experiment. So we did the same exact experiment with ACD15 YFP. We see fast recovery in the wild type, but a significant fraction that doesn't recover in Silencio. And with Silencio itself, we see Silencio CFP recovers very quickly, but much less so in the Silencio H94Q mutant I told you about that no longer interacts with HSP70. So what this tells us is that the Silencio HSP70 activity is somehow regulating the biophysical properties of the MBD6-5-6 complexes at chromocenters, allowing them to maintain this really high mobility. And that is required for gene silencing. So now we've learned something else important about the mechanism here. So now the other big question that we were curious about is what's really driving the concentration of these proteins into these nuclear bodies? And we suspected that it was the ACD proteins. And part of the reason for that is that in other systems, it's known that these small heat shock proteins or ACD proteins can form dynamic oligomeric assemblies in their normal action in protein homeostasis. So at least some of these ACD homologs are known, they're so-called holdases, which are sort of the first line of defense when proteins are unfolded. They grab onto the protein and then they pass it on to the HSP40s, which is another name for the DNA J's. So this would be like the Silencio, and also to HSP70s. And in this process, they're known to form these multimers. Sometimes they can be quite big multimers, like 20-former. There's structures of 20-formers, for example. So Brandon wanted an independent way of testing. Is it really these ACDs that are driving the kind of multimerization? And so for this, he developed what we're calling the SunTag GFP sticky C system. So this is based on an existing Cas9 based system called the SunTag. The way this works is it's a deactivated Cas9. So you can direct it to a certain site in the genome, but it doesn't cut. It just goes there and stays there. And then to that has been fused this chain of 10 small peptide repeats. And then separately, we express a single chain antibody that can bind to those repeats, fused with GFP so that we can visualize this in the nuclei. And then that little sticky C domain that I told you, that's the part that can recruit the ACDs. So theoretically, each of these dead Cas9s would bring in 10 copies of the MBD5-6 complex that I showed you. But if the ACDs are really driving higher order multimerization, then we ought to see something more like this big rat's nest of proteins with lots and lots of GFPs. And we might be able to visualize that as a discrete nuclear body in the nucleus. And so Brandon did the experiment, and indeed we do see these discrete nuclear bodies by confocal analysis. We saw something around 8 of them. Now, some of these are going to be the chromocenters, and that's because, as I told you, these complexes are naturally in the chromocenters, and the sticky C GFP fusion is going to hone in to the natural complexes. And so, and in line with that, many of these nuclear bodies overlap with DAPI staining. But there were a few nuclear bodies per cell which did not overlap with DAPI. They tended to be some of the brighter ones, and we figured those are probably the dead Cas9 sites. And then to really kind of further support that, Brandon redid the experiment in an MBD5/6 mutant. Now, in this case, in MBD5/6, there's no way to tether the ACDs to their natural sites in heterochromatin. So the only place they have to go is the SUNTAG system. And now what we see is exactly 2 nuclear bodies per nucleus, which would be correspond to the 2 homologous chromosomes where the dCas9 sites are. And in line with that, these do not overlap with DAPI staining. And then finally, to really prove that it's the ACDs which are driving this concentration of the fusion proteins, Brandon redid this in the ACD1521 double mutant, and now we don't see any nuclear bodies. We just see diffuse nucleoplasmic staining of GFP, which is similar to a control that we use, SUNTAG GFP TAT1. So what we've learned so far about this complex by these experiments is that it seems like the complexes are driven to high density CG sites by the fact that there are multiple MBDs in these complexes and multimers of complexes, that those multimers are driven, at least in large part, by these ACDs, which can drive multimerization. And then Silencio HSP70 activity is acting to regulate the mobility or fluidity of all of the proteins in this condensate, if you want to call it that, that's sitting on top of heterochromatin. So now this still doesn't tell us why exactly does this cause silencing. And that's something that we are working on now. What we are proposing, and we've got some pretty good evidence now that this is going to be true, is that these higher order multimers of complexes are forming basically a little biophysical environment around in heterochromatin at these sites, and that its function is to keep certain proteins from getting in and allow other proteins to sort of stay in. And we're testing that a bunch of ways, and that's it's too preliminary to talk about that, but we're pretty sure that's at least in large part why this works. Okay, so I wanted to transition a little bit to some kind of technology development that we decided to do that grew out of this project. So we thought, could we use this ACD-mediated multimerization to somehow increase the specificity or efficiency of chromatin targeting? And I'm going to show you a couple examples of what we did. So in a separate project, we've been developing CRISPR-based systems to target methylation to specific loci. And the sort of latest and greatest construct we've developed, which is in the SUNTAG background, we fused together a DNA methyltransferase from bacteria called MP1 together with this plant silencing protein called TRPIP1. And together, this is a very, very potent way of establishing methylation at a target site. The problem is that it's almost too effective because we're also getting lots of off-target methylation throughout the genome. So much so that if you map methylation across the 5 chromosomes, you see that it goes from almost no methylation up to about 40% methylation in these SUNTAG plants. So we're methylating the entire genome. Even more surprising, we're methylating the chloroplast genome, which is normally totally unmethylated. And we're methylating like 80% of the CG sites in there. So these effectors are just so strong that any piece of DNA they bump into gets methylated. So we thought maybe we could use the ACD multimerization technology to make this more specific. So we simply added this little 60 amino acid sticky C domain to the TRBIP1 MQ1 construct, reasoning that this ought to multimerize, just like I showed you Brandon's work earlier. It ought to bring in all the effectors and sequester them away from all the off-target sites. And this worked just, really remarkably well. We essentially completely eliminated the off-target methylation. So now the blue and red lines are the same. The new SUNTAG construct has no off-target methylation. Chloroplast methylation goes from 80% to 0%, and the on-target methylation gets better. So we're really excited about this as a way to make probably any dead Cas9 type application more effective and more specific. The other thing that Brandon tried is adding it to the zinc finger protein to see what might happen. We use the same zinc finger that I mentioned to you that we used in the FWA experiments. And I told you that it binds to lots of off-target sites. Here's a list of the sort of 6,000 top sites that it binds to. So we have this pattern very well characterized. And then Brandon added the sticky C and compared it with a non-sticky C RFP fusion to see what might happen. And what he saw was quite interesting. We get about a 40% increase in the signal of zinc finger binding. There's no change in the pattern, just the magnitude of binding goes up. And we imagine that why this is working is that by multimerizing the zinc finger fusions through ACDs, that we're increasing the local concentration of zinc fingers near the binding site. So when this one lets go, there's another one standing by to jump right back on. And consistent with that, when Brandon repeated this in the MBD5/6 mutant background, which, remember I told you that will untether the ACDs from their natural heterochromatin sites, increasing the concentration that's available for this kind of interaction. Like in the diagram here, what we see is not only bigger peaks, but we see now much wider peaks as well. So we're getting really massive targeting of the zinc finger, and they're all the same sites. We just get much higher targeting there. So we are excited about this sort of approach because if it works with this zinc finger, this may be a way to make any type of transcription factor, silencers or activators, basically better at what they do by bringing more of them to their proper target sites. So we're trying to develop these into some general technologies, and we'd like to get away from the plant proteins and get away from having to use this little sticky C domain, which is essentially just a recruiter of the ACDs. And so Brandon went looking to see are there other ACDs that might work. ACDs are super conserved in biology. They're in all domains of life. And so he's been screening through a whole bunch of them, putting them into the SUNTAG system and looking for ones that form these nice discrete nuclear bodies. And he's found several now that work quite well, as shown here. There are some other ones, including actual human alpha crystallins that work pretty well, but they don't multimerize quite as well as some of them. And then we have some that really don't multimerize at all. So it seems that you have to, kind of pick the right ones in order to work. And then the other experiment that Brandon did is he thought, if the multimerizing is, really the main function of ACD 15 and 21, maybe these other ACDs could substitute for the function of the plant ones in the MBD complex. So what he did is he made some swaps where he deleted the sticky C domain, so you can't recruit the normal complex components, and replace that with a whole bunch of these ACD proteins to see if they might complement. And it worked surprisingly well. So human HSPB1 and 5, and this is one of the I alpha crystallins, they reconstitute the nuclear body formation when they're fused with MBD6, whereas HSPB8, which we had shown in the SUNTAG system back here, did not form any multimers and actually is predicted not to only form dimers. This completely failed to form nuclear bodies when fused with MBD6. And surprisingly, these fusions even partially complement the silencing phenotype. So HSPB1 and to some extent 5 were able to complement the silencing phenotype, whereas HSPB8, which doesn't form nuclear bodies, doesn't complement. So we're continuing to work on these ACD systems. We're trying to kind of develop them into all kinds of different technologies where we can increase the efficacy and specificity of gene targeting systems. I'd be happy to talk to people more about what we're doing there. So that's what I have to say. This is the lab, and almost everything I talked about was Lucia and Brandon. And thanks again for inviting me.